Am I audible and visible? Let me know. Shit.
Unacademy. Let's crack it. Hi everybody, I am Dr. Sharanya from AIMS Patna 2015 batch.
Okay. Am I audible and visible? Let me know, then we'll start. Hello, beta Ashish. Welcome to my class. Okay, fine. Welcome you all. Yes, okay, fine. Welcome to my class. Let us start our class because I'm new here, so I may have some problem in just starting the streaming. Okay, fine. Otherwise, we will, once it gets started, we will be fine. So, hello everyone. Good evening. I'm myself, Dr. Deepthi Karya. I'm MD PhD Physiology. I'm your educator for physiology. Okay. Now, so first of uh, all the questions here at this channel, that is for physiology subject, that is subject in the first year of medical MBBS, nursing, physiotherapy, and homeopathy. So if you are first year student of all these medical faculties, nursing, physiotherapy, homeopathy, and MBBS, okay, these lectures are for you. Here we are trying to complete the whole syllabus of physiology in 1 year in given period of time. Okay, fine. Another thing, all these lectures, they are available as a soft copy in this app, our app Vesalius. It has all the things available. Okay, short questions, short notes, and all. And if you wish to get hard copy, my book is also available, Physiology Notes. So you can order on Flipkart. This is also available. Okay, fine. Now let us start with today's topic. Welcome you all. I think 9-10 students are there, those who are watching me live as well as those who are watching me after streaming of the video. Okay, fine. So now today's topic, first topic, we are starting cell physiology. Okay, I think this you have learned in your lower standards also. Cell, what is cell? Just let okay, cell, how can you define the cell? Let me just a minute. uh, Let me change. I think so. This is the, some of the diagrams are missing in this one. Let me just just give me 1 minute and I will finish this. Sorry for this interruption, because in this PowerPoint I have all the diagrams. Fine. This is the PowerPoint which has all the diagrams now. So here, cell, how will we define the cell? I think all of you are knowing cell is the basic structural and functional unit of the living organism. Okay, cell is basic living unit of the body. Okay, we are multicellular organism, we have multiple cells. Okay, second definition, you can see here, these are our cells. Now these cells, they are collected, they are united, and they form tissue. Okay, so how can we define tissue? Collection of the cells. Very important thing here, word is having same structure and same functions, that is tissue. I think all of you are knowing we have 4 primary tissues. Okay, which are those 4 primary tissues? They are muscle tissue, nervous tissue, then connective tissue, and epithelial tissue. So these are our 4 important primary tissues. Okay, then organ. Now minimum 2 or more tissues, they unite to form organ. You can see here, suppose this is your epithelium. Epithelium, that is covering. Okay, I think you must be knowing columnar epithelium, cuboidal epithelium. So different types of epitheliums are there. Okay, so now suppose you have one tissue is epithelial tissue, and second is muscle tissue. These two, they form your organ, like your stomach. So this is third thing, organ. First is cell. Cell is the basic living structural functional unit of the living organism. Collection of cells having same structure, same function, that is tissue. 2 or more primary tissues, they work together to form organ. Like your stomach, I have given the example. Stomach has got muscle tissue and epithelial tissue. Fine. Then system. Now these organs, you can see one organ is stomach, other is liver, intestine. They form your digestive system. So this is your system. That's fine. Okay, now there are 2 types of cells. One is prokaryotic cell. Pro means before. means before the nucleus, so they don't have nucleus. Example is bacterial cells, they don't have nucleus. Eukaryotes means after nucleus, cells having nucleus, that is eukaryotes, like all our animal cells, they are eukaryotic cells. Fine. So this is our first introductory part to the cell. Now you can see here, this is the structure of a basic cell in our body, entire body. This is the value from Guyton. Our entire body contains, Guyton is standard textbook for first year medical students, as well as homeopathy, Ayurvedic and all. Whenever we are talking about values, always use the values from the standard textbook. So in entire body we have 100 trillion cells, okay, from which 25 trillions are RBC. Now what are the characteristics of cell, which are the important, all the characteristics of cells are the characteristics of life. Okay, one more thing here, try to remember the whole topic when I am teaching, so that becomes easy for you. If you are giving me 1 hour, I feel that you should remember the whole topic. Okay, so now we are enumerating and we are remembering all the characteristics of cell. Okay, number one, so cell characteristics are equal to characteristics of life. Means suppose our body is living tissue, so living tissue and cell is also living tissue, so whatever characteristics are there in our body, same the cell has. Now first is, our body as well as cell requires oxygen and nutrients, so one thing try to remember here only. Okay, second, produces carbon dioxide. Third, it produces energy. Okay. Fourth, it maintains external environment, means it takes nutrient from outside, it gives excretory products to outside. So maintain external environment by working properly. For example, suppose kidney cells are not working, what happens? Creatinine level increases. So the external environment is disturbed. Okay, fine. So when cells, they perform the proper function, they maintain external environment. Okay, next is sorry, sorry, just a minute. Next is, they give response to external stimuli. Okay, whenever there is external stimulus, cells, they respond. And last, very important, all the cells having characteristics of reproduction except neuron. Okay, so try to remember. Sorry, I have throat problem, but no worries, we will do our lecture, we will complete our lecture nicely. Okay, fine. So let us revise all the characteristics. Number one, cell requires nutrient and oxygen, produces carbon dioxide, produces energy, maintains external environment, give response to external stimuli, and reproduction. Okay, so try to remember try to remember all the characteristics. Okay, fine. Now we discuss structure of the cell. Every cell consists of three things. Number one, you can see here, this one, cell membrane. Okay, other names for the cell membrane, that is plasma membrane, you can see, or unit membrane. All the three names you have to remember: cell membrane, plasma membrane, unit membrane. Okay, now what is it? It is a protective sheath that is between, this is extracellular fluid and intracellular fluid. Okay, outside the cell and inside the cell. Fine. Second thing is cytoplasm. Here inside the cell we have fluid part, this is cytoplasm. And third thing, organelle, you can see these are all the organelle: mitochondria, Golgi apparatus, nucleus, endoplasmic reticulum, all. Fine. Now we'll discuss one by one. Now try to remember all the organelles, starting with, this is one, endoplasmic reticulum, Golgi apparatus. Second thing, you can see, I will just show all this organelle also. You can see here, this is endoplasmic reticulum. Just I will sorry. This is endoplasmic reticulum, your this one. Then Golgi apparatus, this is okay. Lysosomes are there. We have lysosomes. These are lysosomes. Okay, peroxisomes, like lysosomes. Then centriole, these are centrioles. You can see these are okay. Secretory vesicles, these are secretory vesicles. Okay. Then your mitochondria. This is mitochondria, this one. Nucleus, all the cells have nucleus, ribosomes, okay, and cytoskeleton. We will discuss one by one each and every organelle and all the structures of cell. Now, sorry. Disadvantage of being teacher. We have sore throat because of our profession. Don't worry, I'll be fine. Sorry. So, starting with the cell membrane. So, cell membrane, you can see here, this is the cell membrane, structure of the cell membrane. It is a protective sheath which covers cell. Okay. Very important thing, this separates, as I told you, extracellular fluid and intracellular fluid. Fine. Okay. Now, very important characteristic of cell membrane is it is semi-permeable. Why semi-permeable? Means it will not allow all the substances to pass. Okay, only few substances they will pass. Now, another important thing, structure of the cell membrane. This is known as fluid mosaic model. This model was given in, if you can remember, this is fine, 1972 by Singer and Nicholson. Okay, they have given this model. Thickness, that is, remember, 70 to 100 angstrom is the thickness, or you can say 7 to 10 nanometer also is the unit. Mainly, the cell membrane consists of, you can see here, lipid bilayer. This is one, this is two. Double layer of lipid. Fine. And these lipids are 40%. Proteins are also there. These are proteins you can see here. And carbohydrates, these are carbohydrates. So mainly lipids are and proteins are there. And carbohydrates are only 5%. Fine. Now we'll discuss the structure. As I told you, there is fluid mosaic model. Okay. Fluid, that is lipid. And mosaic means in between the lipid, you can see here, there are proteins which are embedded. Fine. Important major lipids, remember here, they are phospholipids. Okay, phosphate, it has two parts, phosphate and fatty acid. Okay, fine. You can see here, this is your phosphate part and these are fatty acids part. We will discuss in detail, fine. And other important lipid is cholesterol. We will discuss about first, you remember which lipids are present in cell membrane? Phospholipids and cholesterol, fine. Now, phospholipid, you can see here. This is a molecule of phospholipid. This we have seen here in this previous diagram. You can see here. Multiple phospholipid molecules are there. You can see here. Okay? And they, they form lipid bilayer. You can see here. This is one layer. This is other layer. Bilayer of lipid which is made up of phospholipid. Am I clear to all? Give me a thumb so I can come to know that I am clear. Fine. If you have any doubt, you can write down in the chat box. Your questions are always welcome. Okay, if you have any doubt related to anything, you are most welcome. Fine. Should I continue? Is it fine? Okay. No comment is there in the box. Are you getting me? What we have taught? Yes or no? If no, then also it is fine. If yes, then it is also fine. Just give me a thumb at least. Okay. Waiting for your reply. Now I will continue. Fine. So you can see here, this is phospholipid molecule. This is one. Multiple phospholipid molecules are there here and they form lipid bilayer. Now, we will discuss one phospholipid molecule. You can see here, this is phosphate, this is fatty acid. Okay. So you can see here, lipid substance made up of phosphate and fatty acid. Outer part, this is head part, this one. And this is very good. I'm happy Patel, Parth, good. If you are giving me answer, I'm feeling good that you are also listening and you are getting me, fine. So you can see here, outer part, this is globular end. You can see like globular end, okay, which is also known as head part, okay. and this is tail part, this one, which is inner part. Very important thing here. Outer part, that is polar end. You can see here, and this is water soluble. Okay, and inner part is lipid soluble. Okay, or water insoluble, you can say. So it is outside is water here also inside intracellular fluid is also water, so water soluble part remains outside and inside, okay, in lipid bilayer. Fine. And tail part is here, this one, this is water insoluble. Now these two layers are arranged in such a way that this very important, try to remember this name, hydrophilic. Hydro means water, philic means soluble. And hello Vaishali, thank you, thanks for the reply. And this is, try to remember the name, hydrophobic. Phobia means डर लगता है, तो hydrophobic part, this is tail part. Fine. Hydrophilic, head part, globular part, made up of phosphate. Hydrophilic, tail part, made up of fatty acid. Okay, fine. So head ends, there are two words you can see here, extracellular and intracellular fluid, and tail end are in the center. So this is the structure of our cell membrane. You can see here. Now you can understand the structure. You can see this is phosphate part, this one hydrophilic. Okay, this is fatty acid tail part or hydrophobic part. Very good. Am I clear? Now, in between this, we have this cholesterol. You can see here, these are cholesterol molecules. If you see the diagram, your picture becomes clear. You can easily understand. Okay, so cholesterol. What is the role of cholesterol? Because this phospholipid part, that is soft and oily, you can see. So in between cholesterol, they provide structural integrity. Strength is given by the cholesterol molecule. And second important function here is they pack these phospholipid molecules. Okay. Another thing, the membrane is fluid. What is the role of fluid membrane? What is the advantage of fluid membrane? Because the membrane is fluid, so it becomes, hello Meshwa. So the membrane is fluid. What is the advantage of fluid membrane? Because it is flexible. Okay. So it allows, when it is flexible, what happens? It allows the cell to change its shape without affecting integrity. Means what happens? Suppose something is rigid. Suppose this is flexible and something is rigid. Hello Rushi. So when the thing is flexible, you can easily mold it. Same thing, the cell membrane, if it is fluid, you can easily, the cell can easily change the shape also. Okay, without changing its structure or without getting broken down, without changing the integrity. Okay, another important thing, this is also responsible, cell membrane is also responsible for certain processes. Cholesterol in the cell membrane is also important for growth, for division, also required for secretion, cell movements, and cell junction formation. So these are also the functions of cell membrane. Very, hello Srushti. Other important function of lipid bilayer is it permits this cholesterol in the lipid bilayer is it permits the lipid bilayer to self seal. कहीं पे भी broken down हो गया तो उसको seal कर देगा, healing कर देगा. So that is also important function of cholesterol. Now, function of lipid bilayer. Lipid bilayer, what is the very good advantage of lipid bilayer is because it is made up of fat. Hello Priyanshi. So this fat allows fat soluble substances to pass easily. Okay. So fat soluble substances are our oxygen, carbon dioxide, fatty acid, alcohol. So all these substances, they can easily enter inside the cell, comes out of the cell. It is like there is no cell membrane for these structures. Okay. That is one function of lipid bilayer. Second function, it is a barrier for water soluble substances. Water soluble substances cannot easily pass. Which are the water soluble substances? Try to remember these names: glucose, urea, and electrolytes. So there is a membrane. We have channels, carriers for the you can see this is a channel for the transport of glucose, urea, and electrolytes. Fine. Now we come to second part. So cell membrane, we have discussed cell membrane has got lipids, 40%. So we have discussed, just I am enumerating for the students, those who are coming late. Lipid consists of phospholipid and lipid and cholesterol. Fine. Now this is, you can see in the cell membrane, just I am repeating for the students, those who are coming late. So this is phospholipid molecule, phosphate fatty acid, and this is cholesterol molecule. Fine. And second is protein part. Protein part is 55% and carbohydrate only 5%. Fine. Now protein, you can see these are the proteins. This is channels. Protein forms the electron dense layer because they are negatively charged. Fine. Another important thing, they cover two surfaces of central lipid layer. They are outside also, inside also, and in the membrane also. So three types of protein. Okay. Transmembrane proteins, and you can say that extracellular and intracellular proteins. Now, so this is according to the location, according to the structure. Hello Patel Dwani. Priyanshi, hello. Now, according to structure, we have two types of protein. One that is lipoprotein, lipid plus protein. Okay, and they form channels and enzymes. Channels and enzymes. You can see this is channel. Enzymes are inside the cell. So these are lipoprotein. Other is glycoprotein. Glycoprotein means carbohydrate plus proteins. They form, you can see here, receptors here, glycoprotein. They also act as hormone receptors for the hormone, antigen, neurotransmitter receptors. So this is the function by glycoprotein. Now we discuss according to the location. You can see here, these proteins are integral protein. Integral means, suppose this is this one. Integral. They pass through and through. Hello Mishwa. So they pass through and through the cell membrane. Okay, integral protein. And second is peripheral protein. Either they are outside or they are inside. Like glycoprotein is peripheral protein. Enzyme protein is peripheral protein. Fine. So according to structure, two types of proteins. Let us discuss. One is integral, means inside the membrane. Hello Chaitanya. Chintan. Integral proteins, they are your transmembrane protein. They pass through and through the membrane. Okay, and peripheral proteins are outside. Suppose this is the membrane here, lipid bilayer. Okay, so through and through are transmembrane. Outside and inside they are peripheral proteins. Okay, outside known as extrinsic, inside known as intrinsic proteins. Fine. Now, another important thing, which are these peripheral proteins? You can see here. This are your transporter. Okay, suppose we want to transport glucose or some other substance, we require the protein. So these are your transporters. These are your enzymes. Enzymes are intrinsic proteins. These are your receptors. Here, this one, this is receptor protein outside the surface. Surface identity marker, very important here. I will tell you, blood group antigen A, B and D antigen. These are surface proteins. Okay, cell adhesion molecule, two cells, they fix to each other by the cell adhesion proteins. And last but not least, they help in attachment to the cytoskeleton. So these are all the different types of protein in the cell membrane. Okay, now what are the functional significance? What is the role of all these proteins? You can see here, these are your proteins. This are transmembrane proteins. These are glycoproteins outside. These are the enzymes inside. Fine. Now, important thing here is functional significance of the protein layer. What is the functional significance? Why proteins are there in our cell membrane? What is their role? Okay. Now, we'll discuss one, that is they provide structural integrity, strength to the cell membrane. One thing. Second thing, some of the integral protein, दूसरा नाम integral protein का है transmembrane protein. Okay, transmembrane protein. They are channel protein. Channel means they help to transfer glucose, electrolytes and other substances. They are channel protein. Carrier protein. Carrier means transport of water soluble substances and ions. What is the difference between channel and carrier? channel doesn't have the carrier molecules. Okay, so this carrier proteins are specific for the particular substance. Okay, they are pump, sodium potassium pump, you must be knowing, transport of sodium and potassium ions. They act as receptors. You can see here on the cell membrane, these are the receptors. Act as antigen for the reaction of antigen antibody. They act as enzymes. Enzyme, they act as a catalyst for the chemical reaction. And cell adhesion molecules are also made up of protein. Now let us remember all the functions. Okay, one, structural integrity. Second, channel, integral proteins act as channel, carrier, pump, receptor, antigen and enzyme. 6, and cell adhesion molecule. Now let us revise again. Function of protein: number 1, provide structural integrity. Number 2, integral proteins. They act as channel, carrier, pump, receptors, antigen, and enzymes. And third function, they act as cell adhesion molecules. So this is all. Now next structure in the cell, that is carbohydrate. Okay, carbohydrates. Carbohydrate, as I told you, only 5%. We have discussed lipid, 40%. I am just repeating. Proteins, 55%, and carbohydrates 5%. Lipid में क्या आएगा? What is there in the lipid? Lipids are phospholipids. This is phospholipid layer. This one, phospholipid, and cholesterol. Okay. Proteins, they are lipoproteins and glycoproteins. Okay. And carbohydrates 5% only. They are glycoproteins and glycolipids. Okay. Protein, you can see here, glycoproteins and glycolipids. They are always present on the surface only. So they form a layer. They form a layer on covering the surface, and this layer is known as glycocalyx. Okay. Now, what are the functions? 3 functions. Try to remember. Number 1, because they are negatively charged, so they repel the negatively charged other substances. So carbohydrate, one carbohydrate negatively charged will repel the other carbohydrate, not allow them to enter. Second, if it is positively charged other substance, it will help in the fixation of cell with one another. And third very important function, they act as receptors. Receptors for the action of hormone, for the action of other substances, neurotransmitter and all. So this is about carbohydrate. Okay, now we discuss functions of the cell membrane. Total, all the organelle, carbohydrate, fat, and proteins, all the constituents. What are their functions? Okay, so first, or you can say general functions. One, that is, it is a protective sheath. Protective sheath for all the organelle, because organelle are delicate structures, so they require protection. So first function of cell membrane is it is a protective sheath. Second thing, it will not allow all the substances to pass. So it is selectively permeable. Suppose if it is oxygen, if it is carbon dioxide, it will easily pass. Why? Because it is lipid soluble. But if it is glucose, if it is electrolyte, they will not pass. Why? Because they are not lipid soluble. So it is selectively permeable. Okay. Third thing, maintain shape and size of the cell. Shape and size, because the cell membrane is important for maintaining the shape and size of different cells. And fourth, it links the one cell to the other cell with the help of cell adhesion molecule. We have discussed that is the function of proteins, cell adhesion molecules. Okay, fine. So this is, we have discussed about cell membrane. Now we will discuss about cell organelle. In today's class, we will discuss few cell organelle. Okay. Now, starting with the endoplasmic reticulum, let us enumerate all the cell organelle first. So let us think that we have not forgotten the organelle. Okay. So they are endoplasmic reticulum 1, Golgi apparatus. If you remember, you can write down in the chat box also. Write down the names. Then lysosome, peroxisome. Okay. 5 number, centrosome or centriole. Hello Ram. Centrosome. 6 number, try to remember all the organelle. Fine. 6 number secretory vesicle, 7 mitochondria, 8 nucleus, 9 ribosomes, and 10 cytoskeleton. Fine. Let us remember all. एंडोप्लाज्मिक रेटिकुलम, गोल्गी एपरेटस, लाइसोसोम, पैरोक्सिसोम, सेंट्रोसोम. 5 हो गया. ठीक है. Secretory vesicle, mitochondria, nucleus, ribosomes, and cytoskeleton. We will start with the endoplasmic reticulum. Okay. Endoplasmic reticulum. This is, you can see, complex tubule of series, or you can say that it is a flat vesicular structure. Here you can see this is series of the tubule. You were drawing like this, is it? Fine. Okay. Hello Kishan. Okay, so this is your endoplasmic reticulum. Fine. Complex, याद रखना है, complex series of tubules, and it is a flat vesicular structure present in the cytoplasm. It is near the nucleus. You can see this is the nucleus. Okay, and this structure, these tubules and vesicles, they are interconnected and they form a network. Fine. Very important thing here is this is connected with, you can see, nuclear membrane. This is the nuclear membrane, and this endoplasmic reticulum is connected here. Another important thing, surface area. Try to remember this value also. Surface area can be increased 30 to 40 times when function increases, like protein synthesis, and this increase in the liver cell, because highest function of the liver cell is to synthesize the protein. Fine. Okay, and this is made up of 2 layers, double layer. Inside them there is a lumen, and this lumen is continuous with the membrane of the nucleus, nuclear membrane, and it has matrix liquid that is known as endoplasmic matrix. I will show you another diagram. It becomes easy for you to remember. You can see here, this is double layer. You can see this is nuclear membrane. Hello Chaudhary Saurav. This is nuclear membrane. This is your endoplasmic reticulum. So this is 2D structure, you can see here, and double layer membrane. Fine. Very hello Dr. Sheetal. So this is your endoplasmic reticulum. Endoplasmic reticulum is of 2 types, rough and smooth endoplasmic reticulum. Rough endoplasmic reticulum is also known as granular endoplasmic reticulum. Why granular? It has got granules. You can see these are granules, are nothing but your ribosomes. Okay, and second is smooth endoplasmic reticulum. Here I don't have the diagram of the smooth one. I'll just show you smooth if I have this one. This is the smooth. You can see here no ribosomes are visible. These are ribosomes. Okay, fine. Now ribosomes, they are concerned with protein synthesis. So the function of the rough endoplasmic reticulum, that is protein synthesis. Okay, fine. Now we discuss about rough and smooth endoplasmic reticulum. Rough endoplasmic reticulum consists of, or it is known as granular endoplasmic reticulum. It is due to presence of the ribosomes. Now ribosomes consist of RNA and proteins, ribonucleic acid and protein. What is the important function? Remember here, very important thing, synthesis of the protein inside the cell. For example, which proteins are synthesized? I will give you examples. Like insulin. Insulin synthesis from beta cells of islets of Langerhans. Antibodies from the WBCs. Okay, B lymphocytes, proteins, enzymes. These all are synthesized by rough endoplasmic reticulum. First function. Second function, worn out organelle. Worn out means the organelle which is destroyed. Okay, or they are not working. They are degraded by rough endoplasmic reticulum. So 2 important functions. Try to remember. Number 1, that is synthesis of proteins, and number 2, that is degradation of worn out organelle. These are 2 very important functions of rough endoplasmic reticulum. Next is smooth endoplasmic reticulum. You can see doesn't have ribosomes. Okay, so ribosomes are concerned with the synthesis of protein. Therefore here smooth endoplasmic reticulum is concerned with the synthesis of non-protein substances. Very important function. 2 functions of rough endoplasmic reticulum. Let us remember. Number 1, synthesis of protein and degradation of worn out organelle. Degradation of worn out organelle. Okay. Then next function is storage and metabolism of calcium. Very, very important function in the skeletal muscle. In the skeletal muscle, calcium ion is very important. So this calcium ion is stored and metabolized in smooth endoplasmic reticulum. In the skeletal muscle, smooth endoplasmic reticulum is known as sarcoplasmic reticulum. Remember this thing, sarcoplasmic reticulum. Okay. And third function, catabolism and detoxification of toxic substances like drugs and carcinogens. So three functions. Let us revise. Rough endoplasmic reticulum has two functions: synthesis of protein, degradation of worn out organelle. Smooth endoplasmic reticulum has three functions: synthesis of non-protein substances, storage metabolism of calcium, and catabolism and degradation, detoxification of toxic substances. Fine. Now, so this is first organelle. We have discussed endoplasmic reticulum. Second organelle is your Golgi apparatus. Golgi apparatus, that is made up of series of sac, membranous sacs are there. You can see 5 to 8 sac, or that is known as vesicles or tubules or cisternae. Hello Parth. Okay. Location, this is near to the endoplasmic reticulum. I will show you here in this diagram. You can see this one. This is endoplasmic reticulum. This is Golgi one. Near your endoplasmic reticulum. So whatever protein is synthesized, whatever non-protein substances are synthesized in the endoplasmic reticulum, now they enter in the Golgi. What is the function of Golgi apparatus? Processing and packaging of these proteins and lipids and sort out them. Whether the protein is for enzyme, whether the protein is for hormone, whether it is for other function. Okay, so this is the function of Golgi apparatus. Okay, sorting out of different proteins. Now, this Golgi apparatus, it is located, as we have discussed, near the endoplasmic reticulum. It has two ends. One is C space. You can see this is C space. That is, you can see, near the endoplasmic reticulum, and that is important for entry. Substances they enter, and transphase, through which substances they exit out. Okay. Now, what are the functions? Let us discuss one of the important thing, that is function. So first function, that is, it is for processing of the proteins and lipids which are synthesized by the endoplasmic reticulum. Okay. Second thing, after processing and packaging, what does it do? It causes, processing, it does packaging. पैक करता है, पैकेजिंग करेगा. थर्ड, secretory vesicles. You can see here vesicles are formed. This is C phase, this is trans phase. Now Golgi makes the packages. For example, suppose हम कोई पोस्ट भेज रहे, तो पोस्ट का क्या होगा? पोस्ट ऑफिस में पहले उसका सॉर्ट आउट होगा, फिर उसका डिसाइड होगा कि ये पोस्ट, दिस इज फॉर अहमदाबाद, दिस, और दिस इज फॉर दिल्ली, दिस इज फॉर MP. Okay, so packaging and address. Very good. So processing, then packaging, then sorting out. Fine. And formation of secretory vesicles. You can see here, these are secretory vesicles. They are formed. Okay. Label also. Sorting and labeling also is the function. Okay. Labeling, why? Because we require to know that whether this is the protein for enzyme, this is the protein for hormone. Okay. This is the lipid for some function. Fine. And another very important function is formation of lysosomes. Lysosomes are formed from Golgi apparatus. What is the function of lysosome? We will discuss. So these functions you have to remember. Next, we come to important organelle, that is lysosome. You can see here, these are round, oval, membrane bound. These are also vesicle, because secretory vesicle, these are also vesicle, but these vesicles are specialized type. They have given the name lysosome. Round to oval, membrane bound vesicle. They are produced from Golgi apparatus. Okay, formation from Golgi apparatus. Their size is different in different cells, but usual size you can remember 250 to 750 nanometer. Okay, very important thing lysosome has, it has thick covering membrane. Why? Because inside lysosome is having very strong enzymes, hydrolytic enzymes. Okay, so if the membrane is not strong, what happens? It results in hydrolysis, destruction of the cell. So membrane must be thick. Fine. Now inside you can see the lysosome itself has small granules, and in these granules we have hydrolytic enzymes. Fine. Lysosomal enzymes, these are acid hydrolase. Why acid hydrolase? Because they act best in the acidic pH. When the pH is acidic, then only these enzymes they work best. Okay. Okay. Now, and another important thing, this lysosome and their enzymes are highest in the cells having phagocytic activity. Which are our phagocytic cells? Neutrophils and macrophages. So in these cells, highest number of lysosomes are present. Fine. Now, lysosomes are of two types. Number one is primary and second is secondary lysosome. Now, what is the difference between primary and secondary? Primary means whatever lysosome you can see Golgi apparatus produces. This is your primary lysosome. And secondary means when this lysosome binds with the phagosome. I will show you what is phagosome. Phagosome means you can see whenever this is phagosome. Whenever phagocytic cell it engulfs the particle, they form this kind of vesicle, and this vesicle with the particle is known as phagosome. Okay, phagosome binds with the lysosome. Okay, and this lysosome now becomes secondary lysosome. Fine. Now, and it has various hydrolytic enzymes. Try to remember the names. Easy names are there. Proteases for protein hydrolysis, then proteases, then amylases, lipase, nuclease for nucleic acid destruction, phosphatase for phosphate destruction, phosphate ester, phosphatase. Then comes glycosidase, aryl sulfatase, collagenase. Okay, so these are different enzymes present in the lysosome. Now, functions of lysosome. Number one, that is it causes degradation of macromolecules. Large molecules are destroyed, degraded by lysosome. Second thing, worn out organelle is also degraded by lysosome. This is also the function of rough endoplasmic reticulum we have discussed. Fine. Third function, if the cell is having large amount of secretory product, this is also excreted by lysosome. This is also removed, destroyed by lysosome. And another thing, when the cell completes its life, this lysosomal enzymes, they destroy the cell itself. That is known as autolysis of the remnant of cell, and therefore lysosomes are known as suicidal bag of the cell. So these are the four functions: degradation of macromolecules, degradation of worn out organelle, then removal of excess secretory product, and autolysis of the remnant. These are the functions. Fine. Degradation. Hello, Kishan. Degradation means destruction. Destruction. Okay. It is destruction. Fine. Now, there is applied aspect. As I told you, always write down some clinical aspect at the end of your topic. Okay, disease must come at the end of our class. So here, lysosomal storage disease, what is it? Suppose here if any enzyme, hydrolase enzyme in the lysosome, that is not present or that is not synthesized or inactive, so what happens? This results in certain diseases, and these diseases are known as lysosomal storage diseases. Okay. Now, example, just you remember this thing only: absence of enzyme of lysosome, either they are not synthesized or they are inactive, and that produces disease known as lysosomal storage disease. Example is disease example: acid lipase A deficiency, then eye cell disease, and Tay-Sachs disease. So these are the examples of this. You will learn in detail in your biochemistry. Okay. So this is all about your lysosome. So today we have discussed cell physiology part 1, in which we have discussed cell membrane and cell organelle. They are endoplasmic reticulum, Golgi apparatus, and lysosome. Tomorrow we will discuss remaining cell organelle. So we are ending here. I have completed 45 minutes of my lecture. Okay. Again, for the newcomers, if you wish to get hard copy as well as soft copy of my notes, or you can say of this all topics, soft copy is available in the form of this app Vesalius. You can just download the app from the Play Store and you can subscribe. You can definitely get the soft copy. And if you wish to get hard copy, this is available on the Flipkart and Amazon. So you can purchase physiology notes that is authored by me, Dipti, Dr. Dipti Viren Kadia. So thank you so much. Thanks for watching me. If you like my video, you can like it. You can share with all your friends. I think all my videos will be very much helpful for all medical and paramedical students, all students of MBBS, nursing, physiotherapy, homeopathy, Ayurvedic. I wish that will be benefited. This is my motive only. Okay. Thank you, Kishan. Thank you all. Thank you so much. Okay, subscribe my channel, like the post, and share it with